Sahil, are we good to go? Can everyone hear me?
Yes, I can. I can hear you. I think we're missing. I think one, two, two students. I think one. I think we're good. Everyone's here.
Oh, okay, great. Thank you.
Mhm.
So hello everyone, and my name is Julia, and I'm gonna walk you through some of the supervised analysis tools, machine learning tools that we have on
Hi, Julia.
a platform. Okay, so let me just start from the beginning. So I'm going to be using breast cancer cell line project, and I hope you're familiar with it. Are you? Or should I go over it quickly?
Yeah, Julia, 2 minutes before we actually start the lecture up. I would just take 2 minutes.
Yeah, sure.
So hello everyone. Welcome to today's class. One thing that I really want to mention before we actually start up with the lecture today. So as Ilya promised that he would be sending you guys the second literature review assignment and the other things for this upcoming week. But we did not send it intentionally because we didn't see all the students participating in the Q&A session that was held on Friday at 1 p.m. CST. That's why we thought like, okay, we'll wait till today, let everyone be there in the class and they are clear with today's lecture. Once today's lecture is done, we would be sending out the assignment today. That was the first thing. The second thing is all the videos that have been recorded till now, I mean all the lectures that are there, they all are now available on the educational website. I would be forwarding you the link for the same, so if you have to go back and look to some of the links or like some of the videos, you could access it on the website directly. Let me paste the link over here. Yep, so that's the link. And the third thing is we have built up the organizational pages. So we have a specific organization page for the Georgetown course. So over there, you all would be added to that particular page. So once you guys are added, there would be a functionality of forum. So I mean, I remember I mentioned you to discuss, mention you about this to you guys long back, but now we are all set. The page is done. So you have the forum option, you have the activities option, like activities like something that can update. So we could share everything, all the updates over there, so that it's a mass thing. And you could watch your progress as well, like how much points have you earned till today. I mean, what are the activities that you have done on the server and on the education website. So you could have a look to each and everything. I would be forwarding that link to you guys again today after the lecture. So these are the three things that I wanted to share with you guys. And once again, welcome to the class. And Julia, it's now your turn.
Okay, thank you.
Yeah.
So I'm glad that you're all familiar with the breast cancer cell line project, so we can just begin. So the goal of my talk today will be how to use classifying tools, supervised tools, to identify, in our case, it will be isoforms that can be separate the subclasses of breast cancer. And of course, these tools do not have to be applied only to the expression tables. They can be applied in our example. We will be applying them to expression of isoforms, but in reality, they can be applied to any kind of table where you have some feature some features that actually identify a sample. So it can be mutations, it can be DNA methylation positions, or it can be ChIP-seq signals, any kind of signals that can be, that identify a sample can be used in these classifying tools. And we will be using machine learning methods to identify important features, in our case isoforms, based on their expression profiles. We will visualize what it means that they are important, and we will try to annotate these isoforms. How important are they for the behavior of the cells? So this is the basic pipeline that we will be using. And we will be using random forests that will run multiple times. We will be using LDA, linear discriminant analysis, both as a visualization tool and of course as a classification tool as well. And we will be using decision tree just to narrow down our list of features that can separate the classes. Sahil, I hope you had a chance to send everyone the files. Did you?
Yeah, I did send out the email with the links and with the presentation that you have forwarded me to.
Okay, great. So everybody can follow me. And please feel free to stop me at some point if things get too rush or I'm not clear. I will try and clarify. Okay. So first, just a quick reminder of what we're working with. We are working with isoform expression table, which is a table of expression. And we have obtained it on the platform. It's quite easy. In RNA-seq, let me go for a minute to our platform. If we will press to RNA-seq, we will upload our FASTQ files and we can align them to the known GTF file, to the known genome with bowtie 2 and get an expression table. This was the pipeline that was run. And we got our expression table. Then the expression table was normalized, and normalization is an important topic, and I think you all went through it. This particular table was also standardized. And sometimes we want to standardize the table just because, for example, in linear discriminant analysis, it will be then very easy to interpret the coefficients of the discriminant function, which isoform plays which role. Is it puts in how it separates actually the samples. And so the table we will be working with will be a standardized table. So when you see negative expression values, then the minus sign is there because of the standardization. Of course, expression cannot be negative. Now, the important thing about supervised analysis is that it needs two types of tables. So from our initial standardized expression table, we need to get a test table and a train table. Basically, what this means is that we need some number of samples that the model will be able to train on, that we can build the model on, and we will test this model on another different kind of table. So let me first, the train table. So the train table looks something like this. We have our features, which have to be, for our R script, it has to be the columns. And each feature can be isoform. And we have a class. Now, the class, I will assign each subtype of cancer will be assigned a class, a number. And because this is supervised analysis tool, so supervised meaning that I already know which sample belongs to which class. So I can assign, and for this particular project, we assigned cloud and low was assigned, cloud and low was assigned class number 1, basal subtype was class number 2, luminal class number 3, and normal light class number 4. The test table was exactly the same, except that it's missing a class, because our model has to predict the class of each sample, which is now a row, based on the model. I have here the files that you can find, but I hope that you all can open up the files and see there an Excel file called isoforms Z, which means standardized, and these test and train files. Can you please let me know if you can, if you got them, if you can open them, if you can see them?
Julia, so here's one thing. People are not able to access the links that you have provided, so they need the access to that. So you might have got an email so as to provide them the access. So could you do that, please?
I did give you the files. Those are the same files.
Yeah, but I mean, those are the links to
That's
the
right.
Google Drive. And I mean, when I have forwarded them the links, so they need an access to open those files.
All right. So how do I do that?
You might have got an email saying that XYZ has requested the access. So you could just grant them the
Let
access.
me just stop sharing for a minute.
Yep.
Oh, yes. Okay. Yes. So I Can you try now, please? Is this working? I gave access to everybody. I hope this is working.
Okay, perfect. Guys, please do try it and do let me know on the chat box, or Kian could text me if something is not right again. Okay, I guess Zoo still needs the permission.
Okay.
It worked for Kian. Okay, Bharat needs a permission again. Okay, thank you, Vaishnavi. Okay, Julia, so I guess it's specifically 2 people, Zoo and for Bharat, who need the permission. And for, I guess, I'm not sure who is he or she for Mint. Okay, so Mint needs the permission for other files as well.
For everybody, actually. Is it working now? I think I just made it public, so it should be okay.
Oh, okay. All the three files?
Let me see. Okay, so I need. How do I change this? Sahil, maybe you can give everybody permission? Can you? Or you can't?
Yeah, no, I guess I cannot because this file originates from your drive.
Okay, how about now? Train and the and test also should be okay. Yeah, can everybody see it now? I think I made all of them public.
Okay, perfect.
Yeah, everybody can see.
Yeah, but I guess for Harris, the last file doesn't work. I guess that's the Excel file that he's talking about.
The big Excel file, okay.
Yeah, and the first link still needs the permission. I guess the first link is the same Excel file. I guess that's what Zoo is talking about. So yeah, I guess you could make that file public as well, the isoform file.
Yeah, sure. Here. Okay, sorry. Is this working now?
Yep.
Okay, good. I'm sorry about that. So let me share again.
Perfect. Thank you, Julia. Thank you, guys.
Sorry about that. Okay, share screens. All right. So let's just open the file. And what I wanted to show you is first the Excel file, and just to make sure that everybody understands how we got to the train and test tables. It's not going to work. Great. Okay. Open with Excel. So what I originally had was an expression table. And instead of my names of the samples, I had cloud and low samples, basal samples, luminal samples, and normal light samples. I assigned these numbers to the samples. So I assigned 1 to cloud and low, 2 to basal, 3 to luminal, and 4 to normal light. These are my isoforms, ensemble, coding, I guess. And what is important for train and test tables is this word class. It has to be here. This table is already standardized. That's why you see some of the expressions are negative. And you need to transpose the table. So what you get as an input is this train and test. Now, what are we trying to do? This is, we are trying, let me, from current. Okay, so we are trying to run multiple times, random forest. This will give us, random forest is a classification algorithm, and it's built on many decision trees where you choose each feature multiple times. And you vote how many times this feature has been chosen as a classifier. Then you, our multi-run algorithm will then calculate the frequency that each feature appears. So we have, for example, the same feature here, classifying class I, dividing class A from all the other classes. We calculate how many times and in how many trees this feature has appeared, and we choose the most frequent classifiers, the features that have appeared most frequently. Let's now go back to the platform. We will go to supervised analysis. And we will upload this test and train tables. We upload both tables at the same time. We want to understand which features, which isoforms can classify the samples into classes, into 4 different classes. And we want about 50. We'll take 50 features that have appeared most often. My internet connection is slow, that's why it takes some time, sorry. It will be wonderful if you all can follow me and do the same, so that we will be on the same page, and we can stop and somebody can ask questions if that's okay. So now we want to put the train table into the train group and the test table into the test group, and continue. We will start the pipeline. We have here 2 options, actually, with random forest. We have multi-run random forest, which is the forest we will be using multiple times, choosing the most frequent classifiers. We have the regular random forest, which runs only 1 time, and then you are left with all the classifiers. And we have iterative multi-running random forest, which we might talk about a little bit later after we are done with the regular multi-run random forest. So we put multi-run random forest. It asks us how many trees do we want each time. Just to save some time, let's put 5 trees and 5 iterations. And we will want 50 best features. By best, meaning the ones that appear most frequently. And here, random forest, how do we want to run it? Do we want by the best score? No, we want the frequency. So we want to choose the features that have been chosen by a random forest most frequently. We will use linear discriminant analysis both as a visualization tool to see how well these 50 features separate our classes, and also we might look at the discriminant function for these 50 features. And we sometimes want to eliminate the number of chosen features to only 3 or 4 or 5. So we might sometimes want decision tree. It also helps us, as you remember, linear discriminant analysis gives a linear function that separates all the classes. Sometimes linear separation is not possible, but nonlinear separation is. So sometimes decision tree will be more appropriate. And that's the end of the, and we can name our pipeline random forest. And run. Okay, while it is running, let me just share with you what you will get after that. And what you will get are two important zip files. You will get a zip file for random forest, a zip file for LDA. In my presentation, I forgot to include the decision tree. So you would also get a decision tree. Let's see if the pipeline has completed. No, it hasn't. Is everybody able to run the pipeline as I go along?
Yes, I have. Also in the chat, I don't know if you see it, we're like responding to a lot of the questions because we don't want to interrupt.
Oh, but maybe, oh, yeah. I mean, technology is great, but sometimes it's hard for me to follow where everybody is. Okay, so fine. I'll be happy if you just want to chat and interrupt. That's fine with me. I mean, it will just take a little longer, but I'm okay with it. So if anybody has questions or I'm rushing in, please do interrupt.
All right, we'll do.
Thank you. Okay, so let's see if the pipeline, no, the pipeline is still running.
While it's running, you think you could talk to us a little bit about LDA? Because we've seen the combination of random forest and LDA a lot. And even when we wanted to run the pipeline, that was the only other option. Are they usually paired together like that?
When we wanted to run the pipeline, those were the only options. Well, they are not usually paired together like that. When we built this pipeline, we thought that it will be nice to right away, here, let me just upload these again. They will take time. The idea really was here to run the random forest many times and then to see how well the separation is. And you can see it with LDA. Which is not always possible, I should say, because LDA is a linear discriminant analysis. So it builds a linear function that separates all classes. And multi-random forest or random forest is not a linear discriminant. So you can imagine LDA as a sort of regression analysis. I'm not sure if you're familiar with regression, but it's a line. If you have two classes, it will be a line that separates the two classes. LDA is also, if you imagine two classes, it will be a line separating the two classes. It's just a very easy way of interpreting the results. And it's visualizing the results. So you get a number of features from random forest. For example, we set it to 50 features. And you want to understand, okay, how do these features separate the four classes? You sometimes can do a PCA analysis and see it on the plane. But PCA not always separates the classes well, because that's not the function of PCA. LDA will separate much better. You can also try this TSNE, which is another way of making the of visualizing the data. What this algorithm does, it tries to save the distance between each sample. So when it visualizes the data on a two-plane, on an X and Y axis, the basic idea is to save the distance between the samples. If the samples are in, we have now a 50-dimensional space because we chose 50 features from our random forest. We would like to visualize it in a 2-dimensional space, but we want to save the distance between our samples as was previously. So those are the options, how to visualize the separation of classes. We decided that LDA probably will be the better option just because it's very easy to see.
All right. I don't know about you, but my pipeline just finished recently,
Okay.
the last one.
Yes, mine too. Thank you. So we go to download, and you have here random forest, zip, and decision tree, and LDA. And you have also R scripts if you want to run it with different parameters, change it a little bit, and not use our platform, you can run the R scripts on your own. So we can download the random forest. You can download it on your computer as well. Decision tree and LDA. Okay. Now extract all. Let's start with a random forest. What do we have in our random forest folder? We have this first file, which is a file of how we can open it. Basically, it shows us all the features that we had, and how many times they appeared in the random forest. We remember that we chose 50. So we got to this one. This would be our last one. So actually, this was not a bad decision. But of course, looking at this file the first time, you can, for example, say, okay, I don't really want 50. I want only the very highly abundant or very frequent ones. So I will choose 24 and stop here. And the other files are the test and train files. Let's open the train one. They are not the initial test and train, but only the 50 features are here. If I use here options, formulas, I will see that there are only the 50 features that I chose. I can afterwards use this file, and these are the expression values, standardized expression values of these features, of these isoforms that were chosen as the most frequent separators, as the most frequent classifiers. And I can use this file later, for example, for stepwise LDA, if I want to eliminate some of the features, or I can use that for TSNE to visualize it better. But first, let's see what happened to the LDA and how we can see that indeed these 50 separate the classes. We have in LDA folder, we have this plot. And we can see that the separation is very good. Even on the graph, there are 4 distinct groups of samples. Class 4, 2, 1, and 3. This is if we'll look at LD1 and LD2. If we look at LD1 and LD3, this graph, the separation is also good. Oh, this first one is kind of an outlier. We can also look at the statistics for prediction. And you can see that the accuracy is 1. And so the confusion matrix is ideal. So again, this just shows us that these 50 features that we have chosen as the most frequent ones also linearly separate the classes very well. So let's look at the features. What are the features that we chose? I'm looking now at the train table that is inside the random forest. If I open the train table, this is the table that I have open. I always like to transpose just because it is easier for me to look at this way. What can we say about these chosen 50 features? Well, maybe you're familiar with David, which is a tool for annotation. If you're not familiar, you can visit it right here. Let me see you. You can, or just Google David annotation tool. It looks like this. And we can import these features into David and see what is the annotation of the features that we have chosen. And we will remember that this is transcript, gene list, and submit. Sorry. It did work a minute ago. Thank you. Now, David, I hope that everybody is familiar with David or can follow me. It has 3 options, basic options. You can also, by the way, add some options and play around with it. But the basic ones are the clustering. And the clustering will give us a functional clustering of the genes that we imported. So let's do functional clustering. These are the clusters that we have. And the first cluster is significant. It has a good score, an okay score. It's not very good, but it's an okay one. And a significant Benjamini score for example, up keywords, which is ribonucleoproteins. So we can say that ribonucleoproteins separate, and we can go here, and this will give us, no, not there, sorry.
I have a doubt.
Yes?
What is a good enrichment score if 2.82 is not?
Sometimes you have scores like 24, 25, that would be
Oh, so
as long
larger
as
the better?
Excuse me?
Larger scores are better scores?
Yes, larger scores are better scores. And of course, as long as Benjamini score is significant, you're okay. I would say it like that.
Okay, thank you.
You can see exactly the isoforms that make up this cluster, that make up this group, this cluster number 1, and you can choose them in your table and to see their profiles. How do they separate? So for example, we can choose, because our best class is this ribonucleoproteins, we can read about ribonucleoproteins, of course. This gives us a little explanation, biological explanation of what they do. And this gives us exactly who they are. So we can choose these proteins from our table. We can download the file. There are different ways of doing it. Of course, you can do it by hand. You can do it with Access, just to find these proteins in your table. Right now, maybe it will be just easier to make it, to find them by hand. Find. For example, let's just do 2. This will be easier. And we can filter by their color. And I'm just copying them to a new sheet. It will be easier, of course, if we can order our classes. So let's order the classes. Again, transpose and see their profiles. The profile of the first one, no. Okay, that was interesting. They are very different, these proteins, these isoforms. However, both of them were still chosen as separating. Let us see what happened with the decision tree. Which ones were there? Decision tree. Okay. Lost it. Um, okay. So what can we say so far? Let me come back for a minute to what I wanted to, anyways, to show you. What I wanted to show is that when you have your results of a multi-run random forest, you chose the 50 features, for example. You can annotate them using David. This is playing around with your data, trying the clustering, maybe the table, then trying to separate by the functional clusters your features that were chosen, and thinking up of a biological story. How can these clusters, functional clusters of isoforms, how can they explain the separation into different subtypes of cancer? Because in the end, this is what we are trying to do. We are trying to understand how these 50 isoforms that we have chosen, what role do they play in the cancer cells that separate different cancers into classes? Sometimes the story is hard to imagine. Sometimes this, as we have seen also with our data, the profiles of the isoforms are very different. These two isoforms came from the same functional cluster, but they are not correlated. They are very different. Maybe we shouldn't think of them as one, as doing the same thing in our cells. Sometimes the story of 50 isoforms is so complicated that we want to narrow it down to a smaller number of isoforms. We can use, for example, decision tree or stepwise linear discriminant analysis to try and make the number of the isoforms smaller. So we already ran decision tree. Let us look at the results. The decision tree plot looks something like this. And this gives us a separation, and we actually can see that 1, 2, 3, 4 isoforms separate our classes. And the decision tree separation goes something like a very logical way of separating things. If this isoform is bigger than minus 0.81, we go this way. If it's smaller, this way, and so on. So we can annotate 4 isoforms, and based only on these 4 isoforms, our model is able to predict which sample belongs to which group. So for example, if we have some unknown sample right now of cancer, of breast cancer, based on these isoforms, our model will predict which subtype of cancer this sample belongs to. And again, we can write these down into the David tool, and we can understand the annotation, the real annotation of this isoform. What else does decision tree give us? It gives us prediction statistics, how well this decision tree predicts if you have a new sample, how good will the prediction be? And it is fairly good. It's 85% of the times you will be right. Another possible Another possibility is now to import the 50 chosen isoforms. And do you remember that random forest gave us these test and train tables that already consist only of the 50 chosen isoforms. Let's import them into our pipeline and run stepwise LDA again to narrow the number of isoforms that separate the classes. And I go to random, and here there's test and train tables with 50 chosen isoforms. Train into train group and test into test group. And here I can actually choose a decision tree we have tried. We can choose stepwise LDA. This this parameter tells me how strict, when do I throw out one of the features that is a classifier. Stepwise LDA works as it's an LDA classifier tool that always throws out the classifiers if they are too close to all the others. If they don't add any more value to my prediction, this classifier, this feature will be thrown out. So the stricter I am, the less I will throw out. So I don't want to be too strict. I want to throw out some of my features. But now that we have chosen, with our multi-run random forest, we have chosen these 50 features. Does this mean that actually they are the only ones that classify, that are able to separate our samples into classes? And the answer is no. They probably are not the only ones. And we have seen it also that there are other classifiers that were chosen only once in our random forest. So how would we go about choosing all possible classifiers? Well, we have this iterative random forest, multi-random forest. And the idea is simple. We perform multiple random forests many times, and we always check our LDA function, the prediction, how well the chosen features separate the classes. And we stop when the prediction falls below 70%. So the first time we run random forest, we choose 50 features. Then we throw them out of our basic table, and we again perform random forest multiple times. And again, we choose the 50 features, and so on until the prediction falls below 70%. So if we will do that, we will have all of features that are able to predict the classes. Let's try that. With our initial tables, not only the 50 features, we want to take our initial ones. I have them here. Take some time to upload. For now, we can maybe see if the pipeline with stepwise LDA is finished. Yes, my stepwise LDA is finished. Let me show you what's inside. Again, we have this zip file that has everything inside. We can look at the plot of how the features that were chosen by stepwise LDA separate the classes of samples. And here we see that LD1 and LD2 has separated the third class, which is luminal cancer, very well from all the others. However, the others are very much all together. Let us see the predictions. It seems like statistics. No. Sorry, today the connection is slow. I don't understand why I'm not again. Yes, thank you. Okay, so what I'm trying to see is how well the stepwise LDA chosen features predict the classes. And actually, the prediction is very good. In spite of visually, we are almost not able to separate the classes 4, 2, and 1, at least in LD1, LD2. Maybe here it is easier to separate class 1, class 2, and class 4. The prediction is actually very good, 92%. So sometimes we do make mistakes, but so what is the function that allows this prediction? Let's try to download the function. Coefficients. Let us look at the function. So this is the ID of the chosen features, and we can annotate these. Let's do so in David. We copy them, we go to David, and we upload a new list. It is transcript ID, gene list, and submit. And again, because of my connection, it's very slow. I'm not sure that this is the table. Sorry about my connection. Yes, now David is giving me. Great. We have 6 isoforms that were identified by stepwise LDA as good classifiers, and they predict the class with 92% accuracy. We can look at the table and see what these isoforms actually are. Maybe we can look at the table. Sorry. Your session has ended. I'll try again. No. So while I'm waiting for David to respond, we can discuss a little bit the function. The function, what you see in front of you is one of the stepwise LDA outputs, and you have here you have your isoforms, and these are coefficients of three linear functions. So you can see which isoform has the most impact on each one. So using this graph as kind of a reference point, we can see that the third, or the luminal class, the class of luminal breast cancer, is separated from all others. With LDA, when it is less than -2, you can see here -2, it will be the third class, or luminal. So when LDA is less than -2, so for example, if I multiply all of these, I go back to my table of expressions, standardized, and I multiply each of the isoforms by its expression. By the coefficient, I will get a number, and this number will give me where this sample is on this graph, whether it is up or down. This is actually how this graph has been built. Let me see. No, David is not working properly for me today. Let me try one more last time. Hopefully this time it will work. Now I can see exactly which genes or which isoforms I'm talking about. The first isoform is a transcription factor subunit. So this is phos-like, annexin, collagen, okay, this isoform and integrin. So from our huge table of many thousands of isoforms that identify each sample as a class, we came up only with 6 that, based on these of the profiles of expression of these 6 isoforms, we can predict where each sample belongs to. Or if we have a new number of samples based on this model that we have built, or this is the model, based on this model, we can predict where this sample belongs to, which class. Is it a Claudin-low breast cancer, or basal, or luminal, or normal-like? Of course, there is always a problem with classification algorithms, and the problem is the following. First of all, because it is supervised analysis, we already think that we know how these samples really are clustered. We say that we know that this sample belongs to Claudin-low group, or this sample belongs to luminal class. Sometimes, especially in cancer studies, this kind of speculation is not possible. When we think we know the cancer type, in reality, it can be many subtypes of cancer. Even in our data, if we would do clustering, unsupervised analysis, or clustering of the data, we will see that the samples are not separated by their initial classes, but they are separated differently. So this is problem number 1 with all supervised analysis tools. Another problem that we can run in with supervised analysis tools is the number of overlearning, what is called overlearning in the models. And overlearning is when you have a model that can predict very well where each sample belongs to. However, it is based the model is based on observations that are very close together. So if you give a very different sample, the model will not be able to predict where it belongs to. It is known in literature and from just many observations that random forest, and especially iterative random forest that you do many times, is actually very good with this problem. It does not overlearn. However, linear models, especially LDA, is prone to overlearning. So when you do use these tools, I think it's important to keep in mind all the pros and cons of using different models on your data. So the last thing I wanted to show you for today is really this iterative multi-random forest, where you add up more and more good classifiers to your model. So finally, I have uploaded my initial test and train tables. And I will run this random forest multi-run iterative. Again, it will ask me how many trees I want, and just to save the time, of course, the more trees you have, the better, and the more iterations you have, the better. But just to save our waiting time, I'll put 5 trees and 5 iterations, and each time I want to choose 50 best features. And I want to choose them based on frequency, how many times random forest has chosen them. And maybe while we are waiting for the results, if anybody has questions or something is not clear or anything of that sort, it will be great to hear you. It can't be that I'm so clear.
If I'm going to be honest, I've been following for the most part in the lecture and running the pipelines, but a lot of them I didn't have the same files that you got on yours, and I wasn't sure, but I ran them multiple times, and the same thing happened.
You didn't get the
And
files?
I think also like some of the other kids in the class.
Oh, you didn't get the zip files?
Yeah, so it was.
Can you maybe share your?
No, I didn't.
I will
Sure.
how we can check that. Let me stop sharing. Okay.
All right. Let's see. Is it working?
Yes.
Yeah, so I don't know, like for example, if you go to the random forest lecture,
Mm-hmm.
the one that we did in the beginning, I'm missing there was a third one. I didn't have that one. And then if
You
we
don't
go
have any
back,
of the results. You only have,
the
I think yours
most
failed completely.
yeah. And I ran it a few times too, but I don't know. It was like I did it again here, and I used the same file that you gave us with the Google Drive.
Hmm.
And I don't know. I was talking to a few of the other students and in our group chat, and I think they had
the same problem.
issue. Yeah, but time is we have such little time. I didn't want to like miss out because I can always go back and work on it more after the lecture.
Yeah, but it's I wonder what happened.
If honestly, if I wouldn't be honest, this occurs with a lot of the other lectures, not just with yours. And I think a lot of times, I don't know what the issue is, but it's hard to juggle to run pipelines and listen to the lecture, especially like when it fails and you try to like figure out. But I think
it
you also
will be
have a colleague,
wonderful
so
if
like
you
can
have Yeah, I think it will be wonderful if you have suggestions how to do it better. You know, how to do the lecture. How would you imagine such a lecture? It would be wonderful. From my part, I'm sitting here in Israel, and you know, it will be. Sometimes it's complicated not to see the audience. For me.
Yeah. No, absolutely.
But let me check what happened to your pipeline. Just a minute, and that's what I'm trying to do. And if nobody's pipeline, did anybody get the pipeline? Did anybody get results in their pipelines, or is it just me that's getting the results? With everybody.
Oh no. Seems like it, yeah.
That's pretty bad. Okay. Let me check what happened with the. Right now, it's hard to for me. It's hard to understand what could be wrong with the pipelines. I mean, they're not complicated. You know, they don't take a lot of space or memory on our servers. It's not like running alignments or anything even remotely close. So. So that's very, very. And I'm sorry that it happened. I wonder if Sahil is still here. Sahil, are you here? So actually, you're saying that none of the pipelines ran correctly. That's what I'm hearing. That's what I'm understanding.
Yeah. And it wasn't just this one lecture. I think it's been occurring for like, I don't know, at least for me on like a few of the other ones.
Oh, I'm sorry to hear. Okay. I don't think right now I will solve this, but I will definitely talk to our team about this and try to correct this as fast as possible. But I do see that some people have tried the enrichment analysis or on some other files. Oh. Kian, can you send me the the link to your pipeline, please?
Sure thing.
Thanks. Okay, thank you.
Hey, hi. Sorry, I didn't realize I was on a mute.
Oh,
I was talking, and I mean, thanks, Kian.
okay, Sahil, do you know what could be the problem with the pipelines? I mean, I'm trying to check.
Yeah, I mean,
It didn't work for you.
let me. Might be there is a load on the server, could be.
Hello?
Yeah.
Yep. Hi.
Hi, Leah.
Hey.
Julia, would you like to mention the problem?
Yeah, Ilya. The whole lecture actually turns out that nobody could follow because it didn't work. Nothing worked.
What do you mean?
All my pipelines that I have been running didn't work for any other students. So I'm wondering what, any suggestions? I mean, I don't think I can solve this right this second, but any suggestions that we can do to show anyways how it's done?
Well, do you want to just show your screen, and then once you're done, we can see with Avi what the problem might be?
I did this. I was hoping that everybody can follow me with the files, but I did show and share the screen all the time. I'm not sure that this was easy to follow.
And what's the problem? I mean, what didn't work?
I think nobody got results.
Okay. Yeah, I think we'll have to look into it, but you know, I think we tried last week and it was working fine, at least you know, for what we were trying to do. But definitely, yeah, we should look into why it wasn't working today. And then probably, you know, we'll have the recordings, the recorded session, and hopefully so somebody is saying here that the problem is not with the pipeline. You couldn't get the zip file in the result, so the result wasn't working. Yeah, I mean, we did see. Let me see. I'm just looking at the pipelines right now. I know that last time some people said that it wasn't working, but we looked into it, and there were some simple things that were not set up correctly. So last time, me and Sahil checked the pipelines, and some were not set up correctly. But if today it's different, then maybe we should look into it in more detail. But right now, I'm looking, for example, at a pipeline. Yeah, it looks like people are getting some files, but just not everything. So what I know that if you could please make sure that you don't just look into the final folder. Yeah, so I'm just looking, I think, yeah, Kian's, so let's see. So if you go to download jobs temporary file, or into download all files maybe. Yeah, like for example, I see that for Kian's pipeline, there is a zip for decision tree RPAC and LDA RPAC, but there's no results. Let's see instead. Temp. So let's do this. I mean, you have the pipeline set up. We'll get on this right now, and we will work with Avi to figure out what the problem was. You know, we have our session on Friday. Because of these issues, we didn't want to send out quizzes just on the weekend. I hope that he'll explain that. But as we look into this, you know, we will notify you by email once we've solved the problem. And then, you know, Julia, I think maybe the worst scenario is we'll have to do this again on Friday or next week on Tuesday, if that's okay.
Okay, sure. I hope that it wasn't too bad in any case. And I hope to see you next week anyways, because we will be talking about also unsupervised analysis and other different tools. So thank you, everybody. Sorry.
And yeah, as soon as we have it figured out, we'll get back to you guys and let you know if it's working so you can try this again.
Thank you.
Thank you. So I don't know if you can hear me, but if you could also, I think somebody is recording the lecture. If you could send it out to us, so we can like try to redo some of the stuff, maybe.
Sure.
Yeah, sure.
Oh.
Yeah, I asked Julia to send it to me, and then I can circulate it to you guys as well. And I'll upload it to the education website as well, so that everything is under the same roof.
Perfect. Thank you.
Thank you.